Yeah, I totally misunderstood the sort of way this workshop was put together originally. So as you can see, I expanded my title a little bit. I'm going to talk about sort of a grab bag of topics that I thought this audience might be interested in. I'm going to introduce you to the instrument that we use, the scanning transmission electron microscope, and specifically the 4D variant that I'm going to talk about today. I'm going to give you examples of material science, things that we can measure, properties like local orientation, strain, the phase of the material, etc. I'm going to, of course, you can't give a computational imaging talk in the 2010s or 2020s especially without talking about machine learning. So I'll give you one example on machine learning applications that my group is working on. And then I'm going to move on to even more complicated sort of mathematical inversion algorithms for properties, where we can't just sort of make a simple measurement like how bright the peak is or the position of the peak. We need to do much, much more complicated algorithms to invert the signal that we want. And then I'm going to end with the original topic of my talk, the sort of atomic electron tomography, with some examples in plane wave HRTEM that didn't work so well, and then some ptychography examples that worked much better. So as Paul mentioned, before I can give any talk, I have to advertise for my facility, the Molecular Foundry, Open Access Department of Energy lab. Anybody who wants to can submit a user proposal. So if you see anything in my talk that you're interested in, vis-a-vis electron microscopy, algorithm, code, whatever, please feel free to email us or submit a proposal. So our next cycle is not till March 2023 here, but I also want to mention access is free. Okay, so what is a scanning transmission electron microscope? I wanted to introduce it just in case people in the audience aren't familiar with it. It is one of the most flexible materials characterization platforms that we have. You can use it in imaging mode, where you form a beam, pass it through a sample, and then use a variety of different detectors to measure images, much like we saw in the two previous talks this morning. A huge boon to this has been the widespread deployment of aberration correction. So we can make electron probes now that are below half an angstrom in size. We can probe sort of sub-angstrom dimensions in materials now, and aberration correctors are very much one of the key technologies for that. But you can also operate in diffraction mode. So that's primarily what I'm going to talk about here today. A whole range of probe semi-convergence angles or numerical apertures. But for most of these experiments, we're going to be using pixelated detectors to record full images of the diffraction pattern of the probe. Now, I'm not going to really discuss it today, spectroscopy methods here, but you heard a little bit about them this morning from Hulin, and if you wait later this afternoon, I believe Juan Carlos will be talking about spectroscopic methods in STEM in detail. And then today I will also discuss tomography. Much like you saw in Hulin's talk, you can take any of these methods and then apply it over a range of tilt angles and then measure these structures or properties in 3 dimensions. And so that is a topic I will cover today here. So I just wanted to emphasize a STEM instrument really is like a synchrotron in a microscope, as Mick Brown said. Okay, so I want to focus a lot more on 4D STEM scanning TEM here. We scan our probe across a sample. We used to use single pixel detectors to measure those scattered electrons. But these days, we want to record a full diffraction pattern because it has a lot more information. It's very rich in diffraction information about the sample. So we use a pixelated detector to record a full image of the probe over a grid of 2-dimensional probe positions. And that's where the name 4D STEM comes from. It's a 4-dimensional data set. And it's just sort of a generic catch-all name for the nanobeam electron diffraction or the ptychography experiments that I'm going to show today. So why would we want to do it? Here is the now classic Cornell group paper from a couple years ago in Nature. These are incoherent imaging modes, the single pixel detectors I told you about before, bright field or annular dark field for the unscattered or scattered electrons. And you can sort of make out the molybdisulfide atomic columns. The dark field's a little bit better, but it's quite a noisy image. If you switch to a phase contrast method, in this case center of mass differential phase contrast, you really boost the signal to noise by quite a bit, but you don't boost the resolution. If you look at the Fourier transform on the right-hand side, you can see that our maximum scattering angles that we resolve are twice the numerical aperture, twice alpha of this experiment. But if we do what I mentioned in the previous slide, record a full diffraction pattern, and then do an algorithm called ptychography, we can get both the high signal-to-noise of phase contrast, but also dramatically boost the resolution. And in their most recent paper, the Cornell group has now gotten down to resolution about equal to the vibration of the atom, the sort of thermal envelope of the sample. So the other extreme length scale is very, very large samples. If you had a solid-state battery material, it might be a 10 micrometer stack. We want atomic scale information, but if you did an atomic resolution image, you would need 10,000 of them to cover the thickness of the film, and then another 10,000 images if you wanted some lateral statistics. So we record many, many, many different diffraction patterns from usually about a 1 unit cell size probe, and then we do lots of different analyses in this. So we get all the conventional channels, dark field, virtual bright field, etc. But we could also measure the degree of crystallinity from the Bragg diffraction, the local strain I'll show some examples of here, classify the structures, measure the orientation of Bragg crystals, or even more exotic channels like electron beam induced current shown on the right. And you can imagine for a solid state battery, you might want to know the conductive pathways in these materials. So I'm going to, in this talk, start on the right-hand side here, extreme length scales, and then work my way down to the extremely small length scales on the left. So just as a quick primer, this is a toy model showing what diffraction looks like from a polycrystalline sample. You can imagine this is sort of like a selected area diffraction experiment, where the area we're selecting is the intensity of the probe on the sample surface. So each of these Bragg diffraction patterns is like a fingerprint for the two-dimensional structure of the material under the electron beam. And you can think of this as just a far field pattern or reciprocal space or Fourier space or Fourier transform of the probe convolved with the sample. Now, of course, it's complicated by multiple scattering, overlapping grains along the beam direction, etc. And I'm going to show you some examples in today's talk about how we try and deal with that. I'm not, yeah, go ahead.
Because I am not from this field, so
Please.
I'm going to clarify things. So are we actually getting like Fourier transform, or is this case we're
No, this is a real optical diffraction pattern. So it's not like a Fourier transform of an absolute magnitude. It's like a Fourier transform of the wave function of the electron. But they are mathematically equivalent.
But you're still getting phase information. It's not like you're getting.
No, so these patterns at the end, it's like you take a Fourier transform from a complex field, but we only measure the absolute magnitude squared.
That's what I was asking. Yeah. Sorry, clarify. Yeah.
So many of the algorithms I'll show today will show phase reconstruction too, but generally we can't measure phase directly unless we do something like holography. So I also wanted, I'm not going to discuss it today, but I wanted to show you what this looks like for amorphous materials. If I take a toy model and I randomly displace all the atoms on the right-hand side, and then I do the same thing I did on the previous slide, I scan my probe through it, you can see our nice two-dimensional grid of Bragg spots that represents the reciprocal lattice of this sample disappears, and it's replaced by these characteristic amorphous halos. And this is because there's no long-range translational symmetry in the sample, but there is a first neighbor nearest shell, a second nearest neighbor shell distribution. There are certain atomic spacings that are more probable than other atomic spacings. So you can also use these amorphous halos for a fingerprint of the underlying structure of the material. And this is even worse than crystal samples, complicated by thickness effects, by multiple scattering, because you can imagine if you have a grid of two-dimensional peaks, if the brightness of those changes, you could still measure the spacing of those peaks. But in an amorphous halo, you often completely lose the signal if the sample is too thick there. So let me show you a real experimental example. This is a user sample that they brought to our facility, a pyrochlor single crystal gadolinium titanate sample. They put it next to the reactor, ion irradiated it, you amorphize deep into the material, and then they perform an annealing experiment. You start to regrow from the single crystal into the amorphous material, but you also nucleate a bunch of randomly oriented polycrystalline grains. And so then we do a 4D STEM experiment on it. Here I'm showing 5 probes at once, but we of course only measure a single probe at a time. And you can see as you move from the single crystal to the mixed sort of polycrystalline recrystallization area, and then eventually at the surface of the film, how these diffraction patterns are fingerprints for the underlying structure. So this is an incredibly powerful technique to decouple the step size of the probe from the scale of the information that we're measuring. And so as I mentioned, if I just show you the tableau here, you can easily identify when the probe went through single crystal or this recrystallized fluorite. All the Bragg peaks are in the same locations, but their intensities are different, or the sort of mixed polycrystalline zone or the fully amorphized zone here. So a lot of information is present in these patterns. So I'm going to primarily talk about the analysis of these patterns. I'm not going to talk about the synthesis, sample fabrication, or the experimental data collection very much. My group is very much focused on these data analysis pipelines, so that will be pretty much the rest of my talk here. Okay, so let me just show you some materials science examples. So I already mentioned strain mapping as a possible application. You can imagine if I simply space the atoms out on the right-hand side, push them closer together on the left-hand side, you can already see the reciprocal space relationship here. You get this inverse relationship to the spacing of the Bragg diffracted spots and the spacing of the lattice planes. So all we need to do is perform a precision measurement of this reciprocal space lattice, and we know some information about the local deformation of our sample. So this is that same single crystal pyrochlore data set that I showed before. Here's the strain in the vertical epsilon XX direction, or the horizontal XY direction, or the shear strain, the 45-degree direction there. And you can see the some immediate features that might jump out in epsilon XX, for example, that reddish line, that vertical line, that is precisely where the recrystallization front began to grow. And you can really see there's a big difference in the local strain to the left of that interface, where it was never amorphized, and to the right of that interface, where it regrew on that surface. And here I'm in black masking off the rest of the data set, but we can, in principle, and we have actually done this now, I think, measure the strain for all the different random orientations here. And I also want to call out the infinitesimal rotation tensor theta here. This is the angle in 2 dimensions projected of the strain tensor. And you can also see here, it's all essentially one orientation with a little bit of bending of the foil to the left of the regrowth interface. But to the right of this interface, these grains sort of grow up with about a plus minus 1 degree error on average here. So there's some misfit dislocations at that interface before they start to grow. So another example here. This is an X-ray experiment, spectroscopic ptychography at the cosmic beamline in EELS. This is a collaboration with Will Chu's group over at Stanford. They take their lithium iron phosphate particles, or phosphate, excuse me. These are fully lithiated, and then partially delithiated, and then fully delithiated particles. And they get a measurement of the composition because they do ptychography before the edge, on the edge, and post-edge for lithium. So this is all done with a STEM holder, a transmission electron microscopy holder and sample. We simply take the grid, take it over to NSEM, and then we do 4D STEM experiment on the exact same grid, the exact same particles. And it takes a little bit of doing to locate the same particles in both sides, but now we can make a strain map of the lattice constant of this material. And you can see it's a little bit thick for electron microscopy, but it still works okay. And then pixel by pixel, map this onto the composition. So we're left with a feature vector space here. We have the local concentration, the A lattice, the C lattice. And this is more of a toy model than anything, but it was still a really interesting demonstration. We can learn the chemical mechanical constitutive law, how the local strain depends on the local chemistry directly from the data without inputting prior knowledge. So just a really neat multimodal example that I wanted to show. So the maps before, you can see the particle is about 3 times or 5 times too thick. The diffraction spots look pretty ugly here, and the strain has a lot of error and systematic error in it as a result. So we actually make custom apertures here because we notice that if you change the amount of edge on the probe, you can sub-localize or sub-pixel localize the diffracted disks with much, much higher accuracy. Not just higher precision, but higher accuracy too. So this is just a quick simulation example showing silicon, a thin region and a thick region, with and without a patterned bullseye aperture here. And this was just done using a multi-slice simulation. And what I want you to notice is in the thick region, there's a lot of fine structure inside the disk. That really prevents our sort of correlation position measurement algorithms from working very well. But if you stamp a pattern into the disk, you can use cross-correlation or other simple methods and really get a precise location of those diffracted disks. So I do make these and give them away for free. If you have a STEM, please just let us know, and we'd be happy to send you free apertures for your STEM instrument. But I wanted to show in this cross-validation plot here, this is fitting a lattice to half of the diffracted spots, and then using it to predict the other 50% of the diffracted spots. And you can see that the error, the systematic cross-validation error, has dropped by a huge amount. And so we get about a 4 to 5-fold improvement just by modifying the incident wave function of the electrons. Yeah, so one other example here, moving to orientation. About like a year and a half ago, I realized I didn't fully understand crystallography, and I couldn't write Python code. I had not yet learned to code in Python, so I launched this project to learn both of them simultaneously. So this is a twisted gold, palladium, platinum, or sorry, gold, silver, palladium nanowire. It's sort of this helically grown, twisted nanowires from Mary Scott's group. We did a 40 STEM experiment with a patterned bullseye aperture. Here's what all the diffraction patterns taking the max over probe position looks like. We did our careful, disk detection, masking out of the beam stop, elliptic calibration. Here's the average sort of ring spacing of the first 5 diffraction peaks labeled there. And then what we want to do is solve, we know in this case the crystal structure, but we want to solve the orientation of the different diffraction spots. So our approach is to use a library of diffraction patterns and then fit it in this sort of rotational SO3 space here. And we've explored a bunch of different ways to do this algorithmic fitting, but I'm still interested in talking to possible math experts in the room about this matching in Euler angle space of these patterns. We ended up using a hybrid method where our library is matched to the experimental data using a Fourier transform for the in-plane rotation in polar coordinate space, and then just using brute force numerical taking the maximum in the other 2 Euler angle directions. And that was the fastest method I've been able to devise so far to do this pattern matching. But it works pretty well. Pixel by pixel, we can pull out the orientation of these crystals in 3-dimensional space here. And in this case, we were particularly interested because the hypothesis for how these grow is on 1-1-1 planes, you'll get twinning. So you'll go from one growth direction to another. And so in these orientation maps, what we were looking for is 1-1-1 planes, and more than that, 1-1-1 planes that are shared between 2 adjacent grains, and we were able to find a statistically significant number of those shared planes. So it does look like the hypothesis is correct here.
Colin,
go ahead.
So do you have problems with ambiguous ones? So a lot of times 2 diffraction patterns will look almost the same, especially if you're going down a 1-1-1 axis or.
Oh yeah, we have lots of problems like that. Yeah. So the ways to get around that are to do things like a tilt series, precession electron diffraction, or my group published a paper on multi-beam electron diffraction. We make 8 beams. Those beams intersect the sample at different angles, and that gives you the robustness, right? Because lots of patterns are unique in projection, but if you tilt plus or minus 5 degrees, right, or plus minus 3 degrees, you can find a unique solution.
There's commercial software out there that will tell you that your FCC crystal is filled with single pixel twins.
Ha, absolutely. It's hard to vet commercial software. If only they would publish open source versions of their algorithms, we could vet it as a community, right? Yeah, I'm not going to name any vendors here specifically, but yes. I'm not saying our method's perfect either, by the way. We're still working on this. This, I think I only published this year, so very much interested in increasing the accuracy. And I do have a machine learning project on this, but it's not ready to present to you guys yet. Yeah, so I just wanted to show one other orientation mapping example, ultra-weak diffraction from soft molecules here. Here, we can get basically no diffraction spots except for the pi-pi stacking, the sort of polymer-like backbones of these small molecule soft matter samples. You really have to blow up the contrast to get the diffracted peaks. And this is very much a destructive experiment. We measure the diffraction pattern and then take a 10 nanometer step because we've destroyed all the material under the beam. We have to step far enough into undamaged material to be able to make a good diffraction measurement here. But once we've done that, we can reconstruct the morphology of this soft matter sample. And so here, this is the T1 small molecule without, and with a DIO chemical additive. And you can immediately see in the reconstructed morphologies that I drew above there how different the sort of grain structure of this material is. The one on the left, without the additive, has these long-range, continuously turning pi-pi stacking. And the one on the right, we've really broken up the grain structure a lot. There's a lot of grain boundaries, there's a lot of overlapping grains, etc. And the raw diffraction data is being shown below it there. And so you can see on the left-hand side the sort of slow, continuous rotation of the pi-pi peaks, or on the right-hand side, multiple overlapping peaks jumping as you go across a boundary here. So visualizing these orientations is tough if you have multiple simultaneous orientations. That's why we draw it that way. And just to, again, to make it clear, the old way of doing things with single pixel detectors produces images that look like this. You cannot learn anything about the morphology of your sample. But if you do 4D STEM, you can get a lot more morphological information here. And for my money, my favorite features in this data set is these singularities. If you go around the singularity, the diffraction pattern rotates 180 degrees, not 360 degrees. So this is a plus minus 180 degree disclination defect. And you actually mostly see these in liquid crystals. So I thought they were really cool to observe here in a polymer-like sample. And we're applying this sort of method for many, many other soft matter types of samples. Maybe Andy will talk about that later this week. So one other material science example, just showing the flexibility of the open source codes here. This is a user project from Germany, and the light gray parts are the sort of mostly carbon planes, and the darker gray are the pores of the material that have been filled in with glue. And so they wanted to know what the orientation of the carbon basal plane was with respect to the pores, which are now glue in this sample, but you get the idea. So we take the diffraction patterns, we find all the diffracted peaks, we can fit the highest intensity, and we get the orientation of the carbon basal plane. But if you look at that pattern there, it looks like a whole bunch of noise, essentially, just random rainbow orientations. They wanted to know with respect to the surface of the pores. So I just made a quick mask in Jupyter, and then I differentiated the edges of that mask, and I calculated the normal vector at the boundaries here, and then I simply subtracted the peak orientation from the normal of the boundary to get the relative orientation. And here I've colored it green if it's perpendicular to the basal plane to the pore, and violet if it's 90 degrees. And it's a little easier to see if I blow up that image here. So green is perpendicular alignment, white about 45 degrees, and violet 90 degrees. And you can see it's about 10 times more likely for the planes to be oriented to the basal plane direction here. And so this paper was just accepted, so very happy about that. Okay, so one last thing I want to mention too about just how flexible the instrument is. This is another German group collaboration here. This is Erdmann Spieker's group at Erlangen. And so they didn't have our custom apertures, these tiny numerical aperture probes, but they wanted to make the same measurements. And so they came up with a quite creative way of doing this type of experiment. Normally, when we do a nanobeam electron diffraction experiment, we converge the probe directly into the foil, and then we measure the diffraction pattern in the far field. But if your numerical aperture is fixed to be 1 micrometer, it's very, very hard in most instruments to go to a smaller numerical aperture. So what they do is they basically take a defocused nanobeam experiment, move the sample out of the crossover, and then push the imaging button to go from diffraction space into a confocal condition here. So they are basically refocusing all the diffracted spots into an imaging plane, and when they have a different takeoff angle where they intersect the sample, that means they actually hit the detector plane at a different position, and we can make a measurement of the Bragg diffraction spots here. And so what they call this method SCED, scanning confocal electron diffraction, or 4D SCED here. And if you compare it to the nanobeam diffraction, the main difference is you get sort of Gaussian spikes instead of these circular disks. Why does that matter? Well, it matters if you have extremely beam-sensitive materials. What you really want to do is be able to detect these very, very weak peaks at low dose. You want to concentrate all the intensity into only 1 pixel, something like that, or a couple pixels across. And so the SCED method is much, much better at doing that if you can't have a small circular numerical aperture. And so I just wanted to show the cool results we published here. This is an in situ annealing of this organic film here. This is for an organic solar cell, and they're able to look at the nucleation and growth of these organic crystallites here. So very, very cool experiment. Very different from the polymer example I showed you before, where we destroyed the sample under the beam. Here, they're able to not do too much damage to the sample and actually follow the growth. Okay, so deep learning. I have to talk about it at least a little bit. I'm not nearly as much of a deep learning guru as Hulin, so you should ask your questions of him at lunchtime. But I do want to show one example. So we're talking about diffraction patterns here. If I have nice, well-separated diffracted disks, it's pretty easy to use template matching cross-correlation or one of our other classic image processing methods to find all the positions. So black is the ground truth in this simulation, red is the disks that we managed to detect, and pretty much everything that is like above about 10 to the minus 5 probe intensity we can pick up. But this does not represent most experiments. What if instead we have a much thicker sample, multiple scattering, overlapping disks? If you tilt a crystal to a random orientation, your Bragg vectors might be shorter, and then your circular disks overlap, and they can interfere in incredibly complex ways. So this is what we mean by dynamical diffraction or multiple scattering. Very, very complex diffraction patterns. Our classic algorithms for detecting all the disks do not work at all. They are completely useless. So what we wanted to investigate is, could machine learning help us tackle this type of problem? So what do? We take every single crystal structure out of AFlow and Materials Project and soon Nomad. We simulate hundreds of thousands of different orientations and different crystals. We augment the data. This is what Hulin was really focusing on, how important it is. Random shifts, dead pixels, hot pixels for X-rays, inelastic background, ellipticity of the pattern, Poisson noise, fixed Gaussian pattern noise. You name it, we gotta augment it. And once we've done that, we have this data set of hundreds of thousands of, basically, they're labeled but easy to generate labeled training pairs here, where we have a huge range of thickness of the diffraction patterns, and then we have our projected potentials in 2D, the structure factors of the material. That's our ground truth sample. And then we use a whole raft of different open source tools to do the training on NERSC, to deploy it onto hundreds or even thousands of nodes here. And I'm not going to talk about network architectures too much. I have to say, for problems like this, they're not very architecture sensitive. If you have really good training data, and more importantly, a lot of training data, it will probably work with any network architecture. So the only real two innovations that we have here that aren't found in a standard UNet is we take the pattern itself and then the probe position, and then we do a cross-correlation in Fourier space. So kind of like the first step of the cross-correlation disk detection, and then we move into Fourier space. Now, this is because pooling of information over large distances is much, much more efficient in Fourier space. You pretty much start getting benefits even in the first couple layers here, let alone for a deeper network like the UNet here. And then the other thing we do is every pixel has a complex value in Fourier space and a complex activation layer. So we're using complex RLEU or whatever it's called now. I don't think that's the ideal activation layer, but it works much, much better than using a scalar value for each pixel. And then, as I mentioned, the result is this 2D crystal field sliced in 2D. So here on the left, that seabed pattern, or convergent beam electron diffraction pattern, very easy to interpret. But of course, we include many, many, many diffraction patterns with lots of overlapping disks and confusing contrast, and very thick samples too. So here's just a quick validation to show how well it works. In blue, we're showing the ground truth, and crosses in black, the measured positions, using the old algorithm, correlation disk detection, for 5 of the images in our validation data set, and then just comparing them against our FCUNet deep learning training. And don't pay too much attention to the accuracy numbers. This is the intensity accuracy for the predicted amplitude of the structure factors. But if you look at the positions of the disks, it's basically perfect. It does a very, very good job here. And this is not included in this paper, but we can do multi-objective training too. We can learn not just the amplitude of those diffracted structure factor peaks, but their complex phase, the out-of-plane displacement for some 3D information. This is going to be really useful when we take this network and try and use it to solve unknown crystal structures. More on that in 2023. So I wanted to also make a quick, this is like a couple, like 400 of the data set things here, just to show if you actually try and measure strain from a diffraction pattern, that the SCU net really outperforms the classic correlation method. And maybe even more interestingly than the sort of thickness dependence of the data here is how well it scales with noise. So if you look at the total electron dose, your correlation method only works for detecting the disks when you have about 1,000 counts in the pattern, give or take. But with deep learning, especially because it's operating in the Fourier domain, we can push it down to about 100 electrons per pattern and still get maybe not a good result, but at least a reasonable result. And then on the other end, too, you sort of saturate how much information you can measure in the correlation approach, pretty much around 10,000 to about 100,000 electrons in the pattern. It doesn't get better after that. But the machine learning method keeps getting better until about 10 to the 6 or 1,000,000 counts in the pattern. So it can extract more accuracy, a better scaling law with dose than the classic method here. So far, all I've shown is simulation. It doesn't matter how good it is, though, if you don't apply it to experiment. So I'll just show you one more simulated thing. The first, the motivating example here. Of course, deep learning can do the simple pattern, but it can also do the complicated, difficult pattern as well. So we think that dynamical diffraction, multiple scattering, is fairly easily defeated by deep learning approaches here. So let me show you the experiment. Here's a 100 nanometer thick silicon germanium multi-layer stack. Here's scanning across the sample between the different layers. Diffraction pattern, correlation spikes here, the prediction of our network, and then our measured positions. We measure the spacing of these positions to get the strain of the lattice. And this is the result. So what I'm comparing it to is the EELS composition-based strain. This is epitaxial. So if I know the composition, I know the lattice parameter too. So that's what we've drawn in black here, is this estimated lattice constant. And then in red, that's the network prediction. And blue is the classic sort of convolution algorithm. And you can really see the large systematic errors. And this is because the sample is pretty thick, it's bent a little bit, it's tilted as you move to the left-hand side of the field of view, and the measurement of strain just goes completely off the rails. But the deep learning is much more resilient. And in the perpendicular direction, the strain should be 0 everywhere. And again, the deep learning result is much more realistic. And this result's actually already out of date because we try and routinely retrain this network too, adding more data, more parameter space, et cetera, to continually improve the results. And I should also mention, my group does not believe in software that can't be used by everybody in the literature. So if you use our Pi40 STEM code and you just, instead of calling dis detection, you call dis detection _ MLAI, it will download the network weights and then directly apply them to the data. So we're trying to make it as easy as possible in our Python distributions here for other users to get their hands on this data. Okay, so I just wanted to quickly mention future directions because Hulin talked about it, so I couldn't resist throwing this slide in. The data, of course, that we're recording with these direct electron detectors is out of control. We are getting to the point of petabyte a day. The old way of doing things, the manual approach where you do a trial and error experiment, you make a couple measurements, and then you cherry pick the results, it is just too slow for modern materials science. So we really want to go to an augmented method here, where at all steps, from collecting the data to analyzing the data to even selecting the experiment and running the simulations, machine learning and other AI tools are in the loop here. And of course, the dream is to have the instrument itself, you tell it what you want to know, it selects which information channels it needs to lower the uncertainty, and then it can combine them and then solve for the parameter or the property of the structure that you're after here. So this vision is laid out in our opinion paper that we published last year, if you're interested in reading more about this. Okay, so one last sort of lower resolution 4D STEM topic before I show you some atoms here. This is everything I showed you before, like strain, orientation, etc., those are in some sense simple interpretations of those diffraction patterns. But you can do a lot more if you're willing to build prior knowledge into your algorithms. So I picked out 2 examples here to show you how we do that in these practical experiments here. So the first one is this twisted bilayer graphene sample. You start with a single sheet of graphene, put a second sheet of graphene on top of it, and then twist them with respect to each other, forming a moire lattice. And if I just pause it here, you can sort of see the repeating moire pattern unit cell. So people are particularly interested in twisted bilayer graphene because it really opens up the range of electronic properties that you can have in materials like this, because you're changing the local electronic environment as you move across the Moiré cell. So one of the most famous examples of this is unconventional superconductivity in graphene using the so-called magic angle twisted bilayers here. Now, one thing that we were interested in is if you look at these twisted bilayer Moiré type of films here, lots and lots of different authors, including this PhysRevB paper here, had predicted significant relaxation of the film. And that kind of makes sense, right? If you have AA stacking, carbon atoms on top of each other, it's very energy unfavorable. Graphite has ABAB stacking, right? That's the most favorable configuration. So other authors had predicted, as a function of Moiré twist angle, that you would see different relaxation patterns here as the film tries to maximize the amount of AB stacking. And so we wanted to measure this in an experiment. So if you do classic dark field TEM imaging of these materials, you just throw it on a TEM sample grid. Here is the dark field image, selecting an O1 bar 1 0 type beam, or the other inner shell beams, the 1 bar 2 0 beam. And so you can immediately see in the dark field images, if you look at a single diffraction vector, black is destructive interference and gray or white is constructive interference. So you can see the Moiré unit cell, but not much else. So you can measure the length scale, but we kind of already knew that. We can calculate the Moiré angle, or the Moiré cell size from the twist angle. But in 4D STEM, we get all the diffraction spots simultaneously here. And more importantly, what I've masked off in black here, that's the boronitride substrate. So this is why we're not taking a picture of this in real space. This is on 5 or 10 nanometers of boronitride as a substrate for the device, right? Those diffraction spots are easy to separate in diffraction space because we simply put the graphene down at a rotation with respect to the boronitride. In real space, that's all you would see is the boronitride lattice. So what I just want to show you here is that's what AA stacking looks like, or AB stacking on the two left-hand diffraction patterns. And you can really see, you get sort of the innermost ring is constructively interfering more for AA, and then AB, it's the outer ring, the second ring of diffraction spots. And then saddle point are the 3 edges of alignment here. And so these, again, are fingerprints for the local deformation or local relaxation of the 2 graphene layers with respect to each other. So if we do this, scanning a large field of view, we integrate all of those double overlapping peaks, the diffraction peaks. We basically fit a cosine displacement curve to it. And then this is where it gets really complicated. It's a non-unique solution in many cases. So you have to look at the neighboring probe positions, and you have to do a 2-dimensional phase unwrapping type of problem to pull out what the global displacement is modulo the unit cell of graphene itself. And then if you do that, which we've done, for different Moiré twist angles, this is the result. So it's colored by the displacement legend here, and we're going from about 0.16 on the lower left to about 1.4 degrees on the lower right. White is the extremely favorable stacking direction, the AB or BA type stacking. Black is the AA or BB type stacking, and then the 3 saddle points are colored here in red, in orange, and violet here. And so you can immediately see those models, of course, from many great PhysRevB papers, are correct. The material moves graphene atoms with respect to each other in plane to maximize the favorable stacking. And then as you increase the Moiré twist angle, the degree of relaxation that's possible in these films becomes a lot lower. So if you remember continuum mechanics, strain is the derivative of the displacement field. We have to apply a little bit of smoothing, but we can differentiate these displacement fields and get the strain here. So this is the infinitesimal rotation tensor, unit cell by unit cell after subtracting the Moiré rotation. And I definitely don't have time in this hour to sort of get into the details here, but people can ask me after at lunch. The cool thing that we found is there's sort of 2 reconstruction regimes, whether the Moiré twist angle is less than 0.5 degrees or more than 0.5 degrees. And you can sort of see that in the lower right-hand plot here. Basically, the mode of relaxation, whether you're rotating internally or whether you're shearing at the saddle points, the boundaries of the Moiré cells, that is sort of the 2 ways that the system can lower its energy overall. And so we also confirmed this with a bunch of density functional theory. Very, very interesting study. And of course, I wanted to call out, that's just the first step. So this method, we call it Bragg interferometry, is pretty general. You can apply it to more complicated samples. So in this case, this is a dichalcogenide. materials like molybdisulfide or tungsten sulfide, etc. And we have tried out our method using the same sort of geometry here. 2 TMD layers, a boron nitride substrate, and then we do the 40 stem diffraction pattern. This model is a little bit more complicated. You have to have more interference terms here, but it's possible. We are able to solve the displacement of the 2 layers with respect to each other, and P just means parallel. This is where you have, for example, an ABA above an ABA type of stacking in the material. But what happens if you pick up the material, flip it upside down, and put it back down? Now you have an anti-parallel orientation. Oops. This is where you are going from, say, AB stacking, or ABA, on top of ACA type of stacking. And you get a completely different relaxation mode here. The reconstruction field is very, very different. And I didn't have time to talk about the twisted bilayer graphene. Definitely don't have time to talk about this thing. This is a very, very detailed study here that we're about to submit. So the second example for more complicated extraction of properties when you know strong priors is this STO, PTO, STO superlattice samples. So many people are familiar with this in microscopy, but maybe outside of microscopy you're not. This was sort of first popularized by Ramesh group in this Nature paper in 2016. This is a dysprosium scandate layer and a couple other materials, and then they grow alternating layers of lead titanate, strontium titanate, lead titanate, etc. And what's really interesting about these materials is at certain critical thicknesses, you form vortex polarization domains. And this was a very surprising observation because this is like a nanometer scale control of the domain size in just a typical sort of superlattice multilayer here. And if we plot the displacement of those bright dots, the A-site dots here, you can see the polarization field here. And then in the lower right, this is compared to a second principles phase field type model. So you can predict it in models after you see it in the experiment, at least. So why am I talking about it? We are interested in the 3D structure. So just very briefly, lead titanate is a simple monoclinic cell. The polarization direction, I'm pointing to the left. The A-site atoms, the lead atoms, move to the left, and then oxygen, the counter ions, anions in this case, move to the right. And if I compare that to the cubic phase here, the unpolarized state, you can see how the unit cell has deformed and the atoms inside it have moved. And it's a little bit easier to see if you rotate it back and forth between 0 degree and 45 degree projections here. So at 45 degrees, you can really see if you center on the B sites, the titanium, the lead and the oxygens are moving opposite directions there. So the PTO polarization field can vary smoothly in 3D. What we want to do is understand these domain structures in 3 dimensions. So the domain wall configuration is what we're investigating here. If you look at a dark field TEM in cross-section or in plan view, you get these really interesting superlattice structures. In the cross-section view, it's like the atomic resolution image I showed you before. All the dark sort of dots are vortex domains, right, about 5 nanometers across. But in the 90-degree direction, the plan view, you can see all these boundaries. What are these boundaries? This is what Ramesh charged us to investigate, to figure out what they look like. So if you just look at a single vortex pair, vortex-anti-vortex pair here, and the arrow I'm drawing is the lead shift, so the primary polarization direction of the material, and then red is out of the plane and blue is into the plane, you could, across a boundary, you could go from that configuration to basically you keep the red and the blue the same, but you flip the direction of the polarization on the outside edge. We call this an alpha boundary. Or you could keep the outer edge of the vortex constant and then flip the in-plane axial polarization there. We call that a beta boundary, or you could change both, gamma. And of course, you can go back and forth between the different phases here. Why is this important? Well, it's for electronic properties. The alpha and the beta boundary are what are called chiral domain boundaries. You change the chirality of the polarization field. But a gamma boundary is achiral because you're changing both of the polarization directions here. So we want to measure this. The 3D structure has been predicted, and we sort of know how it's stabilized, but we don't know why or how to measure this asymmetry in the vortex cores, this sort of zigzag, one direction is up and the other direction is down, and maybe even more importantly, the polarization along the vortex core direction is not fully characterized yet. So, and that's because we can only look at the material sort of face on here. What can we do? Well, of course, this is a 40 STEM talk, so we're going to do 40 STEM. The polarization field here, you can see, if I just take a toy model, just like I did before, and I scan back and forth, the diffraction pattern is a good fingerprint of the polarization field. So you can immediately see the left-right polarization changes as a function of the sort of sine wave of polarization I put into the material. So again, if I pause it and we just look at the 200 and 00, or sorry, bar 200 diffraction disks, you can see if I measure the relative intensity of those spots, I can estimate the polarization. And so more detail on that in a second, but I do want to mention this is a pretty thin sample. It's about 8 unit cells thick, give or take. So if you were to do the experiment on the thinnest possible vortex structure, we've sort of float zoned this sample off of the substrate. It's 16 unit cells of STO, 16 unit cells of PTO, 16 unit cells of STO. We're imaging it in the perpendicular direction because we want to see the vortex core polarization. And then this is what the mean diffraction pattern of the experiment looks like. We open up the semi-angles as far as we can without overlapping because we want to have a simple experiment without complex interference here. And if I make a virtual detector on the different diffraction spots that are relevant, you can immediately see this is a vortex domain sample. You can see those long, skinny vortices there. So what we want to do is measure the polarization vector. So if you just do the Friedel pair, the 200 and the bar 200 directions, we can simulate this with block waves. A block wave simulation is pretty straightforward. You just calculate the eigen decomposition of the Schrödinger equation inside the crystal. And I'm going to skip over the math, but I do have these slides here if anybody is curious about it later. And then once you have this is what we call a scattering matrix or an S matrix. And we can use this to very quickly simulate diffraction patterns at different thicknesses. And you can only really use this for unit cell scale calculations, but this is very much a unit cell scale calculation. And so if you wanted to do the S-matrix approach for this STO, PTO, STO, this is why S-matrix is so useful for problems like this. You just multiply the 3 S-matrices together. Very, very straightforward. As a quick side note, all we're doing is taking all the complex operator terms in the Schrödinger equation and then rolling them in to a single S tensor here. And so it just maps the input probe on the right to the output probe on the left. And then, of course, we take the absolute magnitude squared on the left. That's what we measure in the far field on the detector. So I did want to make a side note that simulations, if you're doing a plane wave simulation, are very, very fast if you're doing plane wave, HRTEM style. But we do a STEM probe simulation in exactly the same way. We propagate that probe through the sample. But we need to run another simulation if we take a step, and then another step, another step. So over this square field of view, we're gonna take our 2-second calculation and multiply it by 100,000 probes. So this would be a 58-hour simulation. And one interesting thing that we discovered is that the scattering matrix approach is much, much more efficient. So if you, again, I'm not going to go into the details, but you partition all the scattering angles in the scattering matrix, and then you run the simulation by adding up coherently different terms in the scattering matrix. This is the so-called PRISM algorithm. You get a massive speed up here. So if I just do a single multi-slice scan of that toy model that I showed before, you can go from a multi-slice calculation 58 hours to a PRISM calculation of 4 or half an hour or 10 minutes or even 5 minutes, depending on how much Fourier interpolation you do. Panel I and N and S are pretty inaccurate, so you have to stop somewhere, right? The inaccuracies start to add up eventually, but scattering matrix is very useful for running simulations like this. So back to PTO, STO here. Here are some block wave scattering matrix simulations. And what we're doing is we're varying the polarization from the left from 0 to the right 100%. And if we just highlight just the diffracted disks, the 002 and negative 200 disks here, and I measure the difference, that's what the inset number is. It's not perfectly linear, but it is monotonic with the polarization, even for this sort of thick sample here. But the problem comes in when we go to real samples. If we tilt the sample even a little bit out of plane, so 5 milliradians in the perpendicular direction, you can see our measurements here start to go a little bit askew. We only get 80% of the polarization signal. If we tilt it in the opposite direction, so negative x, and we're polarizing it in the positive x direction, it's even worse. We're only going from a polarization signal of 0.34 to 0.78 here. And that 0.34 is pretty bad. There's no polarization, and yet we're going to measure a polarization induced by tilt of the sample. And it's even worse if you tilt in the forward direction, the same direction you're polarizing. You get signals that range from negative to nearly zero, and then super negative. So it's now non-monotonic with polarization, and it's going the opposite direction. So if we just grid out, red is an accurate measurement, and blue is an opposite direction measurement, and white would be no polarization here. Just as a function of sample tilt, you can see these 4 different cases here. 1, 2, and 3 are okay, but 4, we tilted a tiny bit, and we completely lost the plot here. So this is where we want to make a measurement here. We have all these diffraction patterns. We need some method to invert that signal here, especially for non-orthogonal direction artifacts like that. We want to be resilient against contrast reversals and sign errors here. So there's a couple different ways in the literature. I'm just going to mention Christoph Koch's methods for doing inverse scattering, because he's speaking later this week at this conference. So I'm not going to go into too many details of their methods. Very, very cool, much more accurate than what I'm going to show here, but significantly slower. We have to do this on hundreds of thousands of measurements, so we have to have our inverse algorithm operate very, very quickly. Another approach is this John Spence and Donatelli paper from PRL, also very, very accurate compared to what I'm going to show, but again, much too slow to apply in practice. Our own group's approach using scattering matrix inversion, we published a few algorithms. They might be fast enough, but even here, we're still talking about unit cell scale. And if we want to do full vortices, we're talking about 500 probes times 200 probes, like very, very large fields of view. So what we're going to do is do this flexible inversion using as much prior information as we can. So all we need to do is take our scattering matrix and differentiate it. Again, I'm not going to dig into the math too much, but this is a very flexible approach because all you have to do is differentiate with respect to the variable that you're interested in, and then use the product rule to chain these derivatives through the material. And so we have a derivative for tilt, and we have a derivative for polarization, and then trivial derivatives like the thickness, like intensity or background intensity of the sample. And then from those derivatives, we're going to solve the smallest problem we possibly can. So 16 layers of STO, PTO, STO. Here's our example Jacobians for those 4 tilt cases in the rows that I showed before and the different polarizations. And then from these derivatives here, we can estimate the gradient search direction. So we're going to start with a really, really simple algorithm, alternating direction of minimization. We're going to update our Jacobians, go in the negative gradient direction, add our constraints given by local smoothness or total variation or whatever regularizer we want to use, and then apply any other prior knowledge constraints, like if we know that the median or mean polarization is 0, for example. And then the only 6 variables we're going to measure under every probe position are intensity scaling variables, tilt, and polarization. So as simple as possible, because we just want to get a reliable polarization. So here's the result, or maybe not the result. It's not converged yet in these slides. Steve has the converged values, so you can ask for them at lunchtime or in the break here. It's pretty flat. It's, the thickness of the foil varies a little bit, but it's more tilt of the foil. If you look at the tilt, again, that bears it out. You have a large tilt over the full range. But what we're interested in is the polarization. And so hopefully this is accurate measurements of the PTO layer polarization sandwiched in the middle of STO layers, even with lots of multiple scattering here. So just very briefly, to show you that it does sort of match what we expect. In the axial direction, we expect it to alternate up, down, up, down. So if you go across the vortices here, you expect to go from negative to positive, and it kind of does on the left-hand field of view. You can see it goes blue, red, blue, red, blue, red. The right-hand field of view, though, it kind of is more like white, blue, white, blue. Very surprising to us. So we do wonder if the simple model here is not correct, and we need a more complicated 3-dimensional model of the polarization. The lateral is a little easier. They're sort of, because of the zigzag of the vortices, we expect a net polarization flow in one direction. And so in the type 1 lateral domain here, it should go alternating between 0, positive, which is what we see. And if you go over to a type 2 lateral domain, -0, -0. So it does seem, at least in one of the 2 directions, what we would expect. Okay, so I'm not going to talk about the future directions other than to say, obviously we need more resolution along the beam direction, but there's no substitute for tilting, as you're going to see in the final section of my talk here, the tomography section. Christoph's approach, LARBED, large angle rocking seabed, that's another way to get more measurements, and I actually think that might be the most reliable way to improve these measurements here. And of course, better numerical methods. I'm not a mathematician. I just use whatever crumbs mathematicians are willing to donate. And then, of course, machine learning. So many of these problems that classic image processing algorithms can't handle, or classic mathematical algorithms can't handle. Machine learning is just much more efficient at encoding prior knowledge than any previous sort of method. Okay, so now let's talk about tomography, atomic resolution tomography specifically. So I definitely am not going to go into...
I hate to do this to you.
Am I out of time?
Well, you're a couple minutes over already.
Oh, okay. I'll blast through this then.
Closing in on lunch.
So
maybe a few highlights.
Okay. Atomic tomography, John Miao's group obviously has led the way. Lots of amazing results. If you go to biology, though, they use phase contrast. You get much higher contrast. This is single carbon atoms here. We tried to make this work for plane waves, doing tilt defocus series. The method is called inverse multi-slice. You propagate your beam all the way through. You keep the derivatives at every stage, and so you can run this complex, algorithm in reverse, doing the inverse of the multi-slice operator here. Solve the structure in 3D. It didn't work all that well. This is again an experiment from one of John's students here, or postdocs. We could get the heavy atom sites, but not the light atom sites. It did work when we weren't doing atomic resolution. Here's a clay layer, just showing that we can get good, accurate 3D structures. Oops. Oh. And we even get a hint of unit cell spacing in the clay layers, but it doesn't actually, it gives us what we wanted from this problem, but it seems like plane wave imaging is never going to get us to atomic resolution phase contrast. And so what we did is ptychography. So we scan the whole field of view, we reconstruct the probe and fully deconvolve it from the object wave. And we do this for every single tilt direction, many tilt angles. This is a nanowire sample encased in a double wall carbon nanotube. And we have all the different tilt angles reconstructed in phase. Then we do a class average because we actually couldn't quite get the signal to noise that we needed from the individual projections, and then solve the structure. And here's the resulting reconstruction. And so in the interest of time, I will spend no more than 30 seconds talking about it. It has a two sort of layer structure inside. It has this previously observed zirconium tellurium 5 structure, but instead of in an orthorhombic, like alternating direction configuration, it has this really cool elliptical curve sort of reconstruction on the inner left and the inner right. The other thing we discovered is zirconium tellurium 2, a phase that had never been observed before in this particular arrangement of atoms. But interestingly, the way the atoms are arranged is the same as the zirconium tellurium 5, just without the extra 2 further away tellurium atoms. So we do think it's a realistic structure, and we did confirm it with density functional theory. But I just want to quickly highlight Yongsoo's upcoming talk here. The way forward is obvious. We shouldn't do ptychography and then tomography. We should do a joint reconstruction of the whole process, especially here where we have a large missing wedge. It is very difficult to tilt, as we saw in Hulin's talk, and get a full coverage. And these joint methods are much more resilient against that. Okay, so with that, I'll wrap it up. If you're interested in 4D STEM, I wrote a review paper. All of our codes are available online on GitHub. Specifically, the Pi40 STEM is the code that I used for all of the stuff that you saw in the first 3 sections of this talk. And then finally, our simulation code made in partnership with John's group and one of his former students, AJ Pryor. And with that, I thank my collaborators and would be happy to take questions.